Good morning to all. Myself Praveen Kumar, Assistant Professor in Botany at Chaudhry Shivnath Singh Chandelier, Postgraduate College, Machra, Meerut. Today, you will be introduced to the fundamentals of DNA replication. Now, what is DNA replication? DNA replication is the process by which a DNA molecule produces its own copy. Now, why it is necessary? DNA replication ensures that the two daughter cells formed as a result of cell division receive a copy of DNA. DNA, as you all know, contains the genetic information for a cell or an organism. Now, before we highlight the important points of DNA replication, let us briefly review the DNA structure. DNA is a polymer of deoxyribonucleotides. The DNA is having two strands that are helically coiled around each other. The two strands run in anti-parallel direction, that is, they have opposite polarity. One of the strand is having 5' to 3' polarity, while the other end strand is having 3' to 5' polarity. The two strands are held together by means of hydrogen bonds. Now, these hydrogen bonds are formed between the nitrogenous bases of nucleotides. Now, the nature of base pairing is complementary, that is, a specific base forms hydrogen bond with a specific base only. For DNA, it is adenine that forms two hydrogen bonds with thymine, and cytosine forms three hydrogen bonds with guanine, and vice versa. It is the complementary nature of base pairing that gives an opportunity to DNA molecule to act as template for the synthesis of a new strand of DNA. Now, highlighting the points of DNA replication. Number one, DNA replication is a semi-conservative process. Now, what is semi-conservative? It means that the two strands of daughter DNA, one strand is derived from parent DNA and the other strand is newly synthesized. Means half of the molecule contains parent DNA and one strand is derived from parent DNA and the other strand is synthesized newly. Point number two, the DNA replication starts at a specific location known as origin of replication site or ORI site. ORI site is region 80 base pairs. Point number three, the process is bidirectional. Once the DNA opens up at ORI site, it is having two opening ends that are opposite to one another. The DNA synthesis occurs at both these ends and proceeds in opposite direction. The DNA polymerization or synthesis of DNA follows a specific direction, that is 5' to 3', and it is done by DNA polymerase enzyme, also known as DNA dependent DNA polymerase. The template strand with polarity 3' to 5' shows continuous synthesis of new DNA strand and is called leading strand, while the other strand with polarity 5' to 3' shows discontinuous synthesis of DNA strands and is called lagging strand. Therefore, the process of DNA replication is called semi-discontinuous. Point number 5. DNA polymerase cannot initiate DNA synthesis. A specific enzyme called primase synthesizes a small RNA segment known as RNA primer on template DNA. This RNA primer is then extended by DNA polymerase to synthesize new DNA strand. On leading a strand, only once the RNA primer is synthesized, but on lagging strand, they are synthesized number of times. Later on, the RNA nucleotides of RNA primer are removed by a specific DNA polymerase and replaced by DNA nucleotides. The two DNA segments or fragments are joined together with the help of DNA ligase. The DNA fragments formed on lagging strands are called as Okazaki fragments. Apart from these enzymes, there are others also. Number one, DNA helicase. DNA helicase is responsible for unwinding of DNA. It converts double stranded DNA into single stranded DNA. Once the DNA is unwound, it introduces tension in rest part of the DNA. This tension is relaxed by another enzyme known as DNA gyrase. Once the DNA strands get separated, single strand binding protein attach to single stranded regions of DNA and prevent reannealing or formation of hydrogen bonds between the base pairs. So these are the basics of DNA replication. Thank you.